Welcome to biology. Today we are going to explain DNA replication and put it into very simple terms. DNA replication is one of the basic processes in regular biology in college, and we have to have replication when we have interphase of mitosis. DNA replication occurs. If we have interphase of meiosis, DNA replication. Whenever you need to make more cells for the body, you've got to replicate and make an exact copy of the DNA. This is the process where this happens. Our DNA is 2 feet long, and if it started at one end and replicated all the way to the other, that would be very, very lengthy. So what happens is your DNA opens up in little bubbles throughout that strand and replicates instantly in a very quick fashion. The characters that we want to learn about today are topoisomerase, helicase, the nucleotide as our main character, DNA polymerase 3, DNA polymerase 1, primase, primers, and ligase. Notice many of these words end in ASE, and that gives us the clue that these are enzymes that are going to speed up this process for the body. Let's first look at what is a nucleotide. Now in my animation, my nucleotides are these red characters. Now remember that a nucleotide has 3 components. It is the monomer of a nucleic acid. And the nucleic acid we're dealing with today is DNA, deoxyribonucleic acid. So a nucleotide is made up of a phosphate group, a 5-carbon sugar, and then it's going to have a nitrogenous base attached at the number 1 position. You may be wondering, what is the number 1 position? Well, the core of this lesson is based around this 5-carbon sugar, deoxyribose. The carbons are in very specific orders. The number 1 carbon is always here and always attaches to the base. We have the 2 position, the 3, the 4, and then the 5 carbon is way up here, but that's where it's attached to the phosphate. The rule you need to remember is that the 3 position is always open. It is open for another nucleotide to come in and bond to it. So we read DNA in the 5 to 3 order, and they're referencing the 5-carbon to the 3-carbon orientation. Up here on my board, I have illustrated a DNA strand. This is my DNA strand. You can see I have this side of nucleotides and the complementary side of nucleotides down below. A bonds to T, G bonds to C, C bonds to G, and so forth, and that's what our base pairing rules tell us. A and T always are bound by a double hydrogen bond. C and G are always bound with a triple hydrogen bond. If I want to make another copy of this, I've got to bring in my base pairs to pair up with the existing parent strand. Well, the first thing we're going to do is we've got to unzip this strand. We've got to take the 2 sides apart. We're going to call in our first character, and that is going to be helicase. Now I draw helicase like a pair of scissors because helicase is going to cut and break apart those weak hydrogen bonds. So helicase is going to begin in the middle, which is what we call the origin, O-R-I. The origin is where the replication will begin, and it begins in the middle of your bubble. So if helicase is coming along, unzipping these hydrogen bonds, remember, this is a double helix strand. And so it's actually quite coiled initially, right? DNA is a double helix, and as helicase comes along, this may become very tangled up down here on these ends. So a second enzyme called topoisomerase is kind of like a spinning top, if you will, and it is going to go in front of helicase and get rid of the tangles that might occur. So these two actually work together. moving along, creating the replication fork as we are opening up the DNA strand. So we don't get too many tangles at the end. Let me help topoisomerase do its job. And we're going to untangle that DNA. And we have opened up our strand. So now I've got both of my sides, it's opened up, and I need to make the complementary side to it. I'm going to begin at the origin, and the first thing that happens is a molecule called the primase. Primase comes along and says, I'm going to show you where to start adding the new nucleotides. I'm actually even going to give you a starting flag to tell you to start right at the middle. So here is primase. It's going to say, let's start at the origin. There's your starting flag. And down here, let's start at the origin. There's your starting flag. Well, the first nucleotide comes in and says, okay, I'm going to start here. Remember, they are anti-parallel. They have to run in opposite directions of one another. Notice the carbon positions, 1, 2, 3, 4, 5. So this nucleotide says, this is great. My 3' is open. I can add another nucleotide easily. At the same time, down here on the bottom, this nucleotide has come in, and it says, this is great. This is my 3' position. I can easily add another nucleotide right along behind me. So these two strands add nucleotides easily because the 3' positions are always open. Thus, it happens quickly, and we call those the leading strands. They work just continuously adding nucleotides one after the other, the leading strands. Now, we have the other sides that are open. So let's say this nucleotide comes in and says, well, I want to add here and move in that direction. Wait, that's the 5' position. That position is closed. You can't add a nucleotide there. So, primase says, it's okay. I will show you where to start. I will add another flag for you. Why don't you start adding down here? Here's your starting flag. So it says, okay, well, let me bring in a nucleotide here. Ah, that 3' is open. I can add one to this 3'. I can add one to this 3'. And then we get what we call an okazaki fragment, named after the scientist that discovered this method. Well, I'm not finished. Primase says, that's okay. Let me put down another starting point for you down here. Start adding your nucleotides here. 3' is open. 3' is open. And we bonded all the way. So now this strand is completed, but it happened much slower. So we call it the lagging strand. So we have the leading strand that was really fast, and the lagging strand that was in segments, okazaki fragments. Now the same thing is happening down here, all at the same time. This is all happening continuously at the same time. So primase has put a primer here and says start growing your nucleotides here. There's my 3' position. There's my 3' position, and that works. And then primase says, oh no, add another primer down here. Still that one. And they said, well, okay, you want me to add it here? There is my 3' position. It is open. This one's open. This one's open. Now, as all of this has been busily at work, DNA polymerase 3, which is another enzyme, has been bonding these nucleotides together each time. So if I had 5 hands, I would have been showing DNA polymerase 3, adding the nucleotides in. So that's a character that we need to be familiar with. As you finish and look at your work, you notice, oh no, I still have these starting flags, these primers stuck in my strand. Now, a primer is actually an RNA sequence. And we don't want an RNA sequence inside our DNA strand. So we've got to remove the primers. DNA polymerase 1 comes along, another Pac-Man, DNA polymerase 1, and removes the primers for you. It's going to remove this primer, remove that primer, remove this primer, and remove this primer. So when that's over, you notice, well, I've got these spaces. Look at these big gaps left in my strand. And our last character, which is kind of like a glue stick, and it is called ligase. Ligase comes in and glues our fragments together. And even glues the adjacent bubbles together. At the end, you have 2 different strands. This is called the semi-conservative method because one strand is the parent strand and this is the new strand. So I have this DNA strand, it is identical to this DNA strand, and they run anti-parallel. I hope you've got DNA replication and now you can speak the lingo.